Okay, now let's go to the next main topic, the eukaryotic cell cycle and mitosis. So in our discussion, we will be limited to the cell division of eukaryotes. Most of the discussions involving prokaryotes will be reserved in your microbiology classes. So, okay, eukaryotes. As a review, eukaryotes are those organisms whose cells have a membrane-bound nucleus and other organelles enclosed by a membrane. In contrast to prokaryotes, they would have larger cells and longer gene sequences. And as an adaptation, the long gene sequences of eukaryotes are compartmentalized in multiple chromosomes. So before we proceed, let's just differentiate some related terms. So let's just try to differentiate gene from DNA, from chromatin, and from chromosome. Okay, so let's start with DNA. So when we say DNA, it is, okay, so let me just write that, DNA. So when we say DNA, it is a molecule that contains instructions needed for the organism to develop, live, and reproduce. So in terms of structure, this occurs as a double-stranded molecule. And this molecule would contain those instructions. So think of it as a cookbook that would contain instructions separated into chapters. Now, when we say gene, it is a portion of the DNA. So let me just draw. It is a portion of the DNA stretch that encodes the synthesis of a product, such as structural proteins and functional RNAs. So if we will go back to our cookbook illustration, DNA would be the entire cookbook, gene would be a particular chapter that would contain instructions to make a particular product. And that product can either be or can be a structural protein or functional RNA. Okay, next is chromatin. So what is chromatin? Basically, when we say chromatin, it is a condensed and compacted DNA plus proteins, especially histone proteins. And so this DNA stretch would be condensed and compacted, and with the addition of some proteins that will now be called as chromatin. So this chromatin will further coil and condense, giving rise to the chromosome. And this chromosome will reach its max condensed state at the metaphase stage of cell division. So in the metaphase stage, chromosome is seen as the crisscross structure or butterfly structure that align in the metaphase plate. And so chromosome occur either as duplicated, no, as seen in the metaphase, this conformation, this is a duplicated chromosome, or it can occur as a single chromosome, as seen in the anaphase stage of cell division. Now, this chromosome is a subpart of the organism's genome. And when we say genome, this is the complete set of genetic instructions of an organism. So with these terminologies cleared, let's now proceed. As mentioned, chromatin is a condensed DNA molecule plus proteins, primarily histones. These proteins maintain the chromosome structure and control the activity of its genes. So this chromatin will further condense into, that's right, chromosome, and it's the chromosome that will participate in the karyokinesis, or separation and segregation of the genetic material into the new daughter cells. So chromosome, that would be the highly compact genetic material, and is visible with a microscope. Okay, so the chromosome seen in the late stages of the prophase would assume the duplicated conformation. So when we look at illustrations of chromosomes at the prophase or late prophase stage, it looks like this. So this is actually a duplicated conformation, and this chromosome, or this portion, is the exact copy of this portion. These copies are called sister chromatids, or to be more accurate, twin chromatids, because they're genetically similar. So this whole structure is a duplicated chromosome, but if we just focus on one member of this duplicated chromosome, it's called a chromatid. This is a chromatid. This is another chromatid. Taken together, this is a duplicated chromosome. And this duplicated chromosome, or these sister chromatids, are joined together by a structure called the centromere, the narrowed waist. Okay, so when the cell divides, the chromosomes, the duplicated chromosomes, align at the metaphase plate. So this is an imaginary plane at the equator. The chromosomes would align at the metaphase plate and undergo a type of cellular tug of war, where each sister chromatids are separated and pulled into opposite poles. So we have here a chromosome. It will undergo cell division. So particularly if we're in the anaphase stage, the sister chromatids will be pulled apart. So one chromatid will be pulled to one pole, the other to the opposite pole. And when they're separated, they are no longer called as chromatids. They are now called as chromosomes, single chromosomes. So these single chromosomes will eventually be sorted into the new daughter cells. Okay, so in this figure, you can see chromosome duplication and distribution. So here on the left, this just shows you an electron micrograph of a duplicated chromosome, the crisscross structure that I'm talking about. So this whole structure is a duplicated chromosome. This portion is your sister chromatid. This is another sister chromatid. But again, taken together, it's a duplicated chromosome. So, so when the duplicated chromosome undergoes anaphase, these two members would be separated. And once separated, they are now called as single chromosomes. So this figure also shows the same thing, the duplication and distribution of chromosomes during cell division. Okay, so now let's go to the phases of the cell cycle. The cell cycle can be broadly divided into these stages. First is the interphase. So this is the preparatory phase, and this is where the cell contents are duplicated. And so under the interphase, you have substages. First, you have the G1 phase. So this is the phase where growth happens. So there's an increase in the overall cell contents. So there is a doubling of the number of organelles and other cellular contents. Next, it will be followed by the S phase or the synthesis phase. So here is where the chromosomes are duplicated. And then next we have G2 phase. So growth 2 phase or gap 2 phase. This is the stage where the cell undergoes further growth and does other preparations for the actual division for the next stage. So preparations such as checking of replication errors and needed repairs. So next is the division phase. And so for the division phase or the karyokinetic phase, it can either be mitosis or meiosis. But for this part of the lecture, we will focus first on the mitotic phase. So on the division phase. So in this phase, this is where the actual separation of cellular contents would occur. So of course, you have the mitotic process. So this will be the stage where the nucleus or the genetic material would be divided and segregated to two new daughter cells. And then it will be capped off by cytokinesis, which is the division of the cytoplasm. Okay, so this is an illustration of the cell cycle showing the duration, relative duration of each process. And so as you can see, interphase would consist the bulk of the cell cycle, with the M phase or the division phase being a short period phase because of the transitory nature of its substages. So kung makikita ninyo, mas mahaba yung oras na kailangan sa pagpo-prepare kaysa doon sa actual na hiwalayan. Okay, so now let's just zoom into the specific karyokinetic process, which is the mitotic process or mitosis. So as defined here in this slide, mitosis is the process in which a eukaryotic cell nucleus splits into, followed by division of the parent cell into two daughter cells. So an important feature of mitosis is that the ploidy, or the number of chromosomes of the resulting cell or of the daughter cell is similar to the parent cell. So what we're trying to say here is that if your parent cell is 2N or a diploid, after mitosis, the daughter cells would also have a ploidy of 2N. Okay, so just to illustrate, let's just say we have a cell with a ploidy of 2N is equal to 4. Okay, so what does this mean? Okay, so let's discuss this part by part. Okay, so the number on the right is the total number of chromosomes. In this case, 4. So we have 4 chromosomes. Sorry, we're talking about mitosis, so they're duplicated. In the note, so we're clear about the number on the right. The notation on the left is the number of sets. That the number of sets of chromosomes that the organism has. So in this case, 2N would mean this organism would have 2 sets of chromosomes. And for organisms with 2 sets of chromosomes, they are called diploid, or this state is called the diploid state. And if we will try to deduce the number of chromosomes per set, so mathematically you can easily get per set there are 2 chromosomes. So for diploid organisms, usually these 2 sets are the maternal set and the paternal set. And so if we will try to illustrate this cell, you have 4 chromosomes in total. You have 2 sets, 1 maternal set and 1 paternal set. So let's just say the red would be the maternal set, and then the blue would be the paternal set. And each set would have 2 chromosomes. So for the maternal set, you have chromosome 1. Also in the maternal set, you have chromosome number 2. And on the paternal set, you have chromosome 1. And then also on the paternal set, you have chromosome 2. Okay, so in the metaphase stage, these chromosomes, these duplicated chromosomes, would align at the metaphase plate. So they would look something like this. So let's just try to alternate, change color. Okay, so in the metaphase, they would look like this. And then in the anaphase stage, as we've mentioned before, they will be pulled apart. Okay, so what will be pulled apart? The sister chromatids. So this portion, okay, let me just change the color. So this portion, or these chromatids will go to this pole, and these chromatids will go to this pole. So when we enter anaphase, it would look like this. Okay, so they will be pulled to their respective poles, and at this stage, these structures, sorry, these structures are now called single chromosomes. They are not chromatids. They are chromosomes already. And at the telophase, of course, after telophase in cytokinesis, it would result to two cells with haploidy still similar to the parental cell. Okay, so these are the two daughter cells. Daughter cell number 1, daughter cell number 2. And as we have defined from the previous slide, after mitotic process, the daughter cells should have the same genetic composition as the parental cell. So after mitosis, your daughter cells should still be 2n is equal to 4. Okay, so let me just clear the board. Okay. And so let's check if the ploidy of the daughter cells is still 2n is equal to 4. Okay, let's just examine. Okay, so as you can see, they have the same. These two daughter cells would have the same composition, but let's check their ploidy. Okay, so is this still 2n is equal to 4? Let's check. 2n is equal to 4. Is the total number of chromosomes still 4? Let's try to count. 1, 2, 3, 4. 1, 2, 3, 4. Yes. Next, how about the number of sets? Is it still composed of 2 sets, one maternal and one paternal? Yes. You have paternal set 1, maternal set, the red 2. Similar to this. You have a maternal set and then paternal set. And then each set, do you still have 2 chromosomes per set? Yes. For the maternal set, the red, you still have 2 chromosomes, 1, 2. Paternal set, 2 chromosomes, 1, 2. Same with the daughter cell number 2. Okay, so as demonstrated, the ploidy of the parental cell is conserved up to the 2 new daughter cells. It's just that for the new daughter cells, the chromosomes are already in a single conformation in contrast to the duplicated chromosome that is seen in the parental cell. Okay, so let's just continue our discussion. Yeah. So mitosis progresses through a series of stages. We have the prophase, metaphase, anaphase, and telophase. Sometimes there's a transitory stage between prophase and metaphase called the pro-metaphase. But mainly, literature would discuss or highlight these four: the prophase, metaphase, anaphase, and telophase stage. And actually, we have somehow discussed or demonstrated this from the previous slide. And so this just shows you an illustration, or this picture just shows you one of the earliest illustrations of mitosis. Okay, so as for this illustration, it just shows you the events in the mitotic phase and the corresponding processes that happen in each stage. So right before the mitosis, you have the G2 of the interphase. And so what happens here is that, as we've mentioned before, this is a preparatory stage. So in the G2 stage, the genetic materials should have been duplicated already and have been checked before the actual separation, actual karyokinetic process. So next we have the prophase. So prophase, what are the main events there? Chromosomes are already present. Take note, chromosome, not chromatin, because the chromatin in the interphase should have condensed into a chromosome. Yeah. So in the prophase, chromosomes are present, and the nucleus is tightly coiled. And as you can see, the spindle fibers are beginning to form. Next, you have the prometaphase. For the prometaphase, what are the events there? And so you have the nuclear envelope breaking. And then next, you have the spindle fibers extend from the nuclear region to the poles. Yeah. So this would, in effect, they would fully stretch the spindle fibers. And then next, the spindle fibers would also attach to the chromosomes via the kinetochore. So when we say kinetochore, it's just that disc-shaped structure in the chromosomes that serve as point of attachment of the spindle fibers. Okay, next we have the metaphase. So for the metaphase, the mitotics, as you can see, the mitotic spindles are already fully formed. The chromosomes would align at the metaphase plate. And the kinetochores of the sister chromatids would face the opposite poles of the spindle. So of course, this is in preparation for the next stage, which is the anaphase. This is the stage where the sister chromatids would separate at the centromeres. So this is the cellular tug of war. So the sister chromatids will be separated. And once separated, these structures are now called single chromosomes or daughter chromosomes, which will move to the opposite poles. And that will prompt the next stage, which is the telophase. So for the telophase, what happens there is that the cell would continue to elongate. The nuclear envelope reforms around the daughter chromosomes. The single chromosomes would start to uncoil, and then the mitotic spindles would also disappear. And then cytokinesis. For the cytokinesis, it's the actual separation of the cytoplasm. So after cytokinesis, you will have two physically separate daughter cells. Okay, so let's just look at the mitotic spindle. So basically, it's just a cluster of microtubules that would be important in the actual migration or movement of the chromosomes. So these would be the electron micrographs showing the spindle fibers. These thin thread-like structures here are the spindle fibers, and these would be the chromosomes. The thicker lines seen in this micrograph would be the chromosomes. Okay, and now in the final stages of the cell division, of course, there will be the actual separation of the cytoplasm, and we have emphasized this already. It's called cytokinesis. In the process of cytokinesis differs in animal and plant cells. Recall that animal cells have more flexible and pliable plasma membrane as its outermost covering. So for animal cells, their outermost covering is a plasma membrane. So because of this characteristic, the division of the cytoplasm can occur by simply pinching off, because you don't have the limitation of having a rigid outer covering, which is the exact uh, issue or concern for plant cells. For plant cells, they would have a rigid cell wall as their outermost covering. So they also have a plasma membrane, but um, outer to that region would be a cell wall. And cell wall would have a cell wall is a rigid structure, and hence your cell can't just simply pinch off. Okay, so first let's just discuss the cytokinesis in animal cells. So for the animal cells, this would be the process. First, there's a formation of a cleavage furrow. So yeah, a cleavage furrow forms from a contracting ring of microfilaments interacting with myosin. And then after, so this would be your cell, there would be a cleavage furrow. And eventually this cleavage furrow would just deepen and would eventually divide the cell into two separate cells. Okay, so this is a better illustration. Now for the plant cells, there's an additional step, or it's quite different because as we mentioned before, the parent cell can't just pinch off into two cells because of the rigid cell wall. So for plant cells, these would be the steps for the cytokinesis. So first, there's a formation of a cell plate in the middle. So this cell plate is formed by vesicles that contain cell wall material. Later, or on the next slide, we have a better illustration of this. And then next, after that cell plate formation in the middle, that cell plate, that very cell plate, would grow outward to reach the edges and would physically divide the contents of that single cell into two cells. And then after that, each cell would now have a plasma membrane and cell wall. Okay, so those steps are illustrated by this figure. Okay, so first, you have, so this is the parent cell, and then the genetic material has already divided into two daughter cells, and then now cytokinesis should ensue. So for the plant cells, cytokinesis would take place like this. First, you have the formation of cell plate. So you have vesicles that would move towards this region. And then these vesicles would just coalesce or unite to form a solid cell plate. And then this cell plate would just extend to reach the edges of the cell. And once it reached the cell wall, it would effectively divide your cell into two new daughter cells. Okay, so in this slide, this is a section of an onion root tip. So can you spot the different stages of mitosis in this slide? This is actually a good or a beautiful slide showing mitotic process.